this is our IRP video number 1 for Manny and I. We're doing the GMOs and testing to see what different organisms, or not necessarily organisms, but what different samples you can test to find out if there is a genetically modified organism or genetically modified gene found in those samples of food. What we were doing this week for our experimental plan was we were going to confirm what sequence we were going to look for in our samples, and we were going to use that specific sequence to detect if that, or use that modified sequence to detect if those foods have been modified in any way. Along with that, we are also going to design and get our primers for that sequence, and then we're also going to pick some samples. What we were able to accomplish this week is we were able to decide that we were going to go with the cauliflower mosaic virus, which is what that previous group used to do the same project. So we're going to use their primers is what we've decided to do. Rather than make our own, we're going to use some that have already been proven successful in this lab setting. So we want to do that to try and recreate, but also find some new samples that possibly have genetic modifications to them. Other things that we accomplished this week was that we decided what kind of samples we were going to use. We're going to do some things like fresh corn. We're going to do things like Doritos, canned corn, popcorn, hominy, even vitamin D milk. We looked that up, and it supposedly has corn products in it. And so we're going to test that along with Honey Bunches of Oats and some other different samples that we're going to use for this project. So what individual contribution I had for my group this week is I helped come up with some samples that we would be able to test, and also some how we could modify our schedule a little bit, because we are at this point with the days that we have, we're maybe a little bit behind schedule, especially having to wait till Tuesday for our primers to show up, because PCR is the first thing that we're going to have to do after we isolate our samples, our DNA. So we talked about how we'd be able to improve that and be able to then perform isolation of our DNA on Tuesday, and then be able to run some PCR on it. That way Wednesday we'd have some time to be able to perform some of our experiments and be able to get the ball rolling on that. Experimental challenges that my group faced this week, just designing primers. Luckily that group had those primers designed already, so we're able to use those, which was a big help. Along with that, as we were looking through our protocols, we found that some of the volumes and concentrations of things like our primers weren't very specific. So that was a challenge that we faced, because that's a big part of PCR, is making sure you've got the right concentration of primers so you don't have primers forming to themselves, but also so that you've got enough to attach to all the DNA that you're going to be making so that your enzymes can function and work properly. For me, what insights I helped to provide or help fix those problems, brought up with Manny how we'd be able to use different concentrations at first, and for our first sample PCR, rather than making up our whole sample, Sister Walker mentioned that we should do some test runs. So me and Manny, we talked about how we could do some samples that start with concentrations that we've used in the lab, so around 10 micromolar, and reducing that down or having a little higher for our DNA samples, so that when we run our PCR, we can have multiple samples first, so we can then run them on a gel and then see what ones worked best. And then we don't waste product, and we don't waste other things on samples that won't work. So those were things that I brought up, and those are things that we're trying to get through. And so we'll hopefully, once we get our primers, we'll be able to get the ball rolling on all of this and be able to get ahead of the game and be able to get our samples and isolate some DNA and find out which things are containing modified corn and which things don't.